So the main research focus of the Vienna Airway Lab is type 2 immunity of the upper airways. We are focusing on respiratory allergies, as well as chronic rhinosinusitis, and there especially on the subtype with nasal polyps. And we are also working together with the Department of Pulmonology to address the question of the United Airways.
Because we're using patient-derived cells, it's very difficult to find an ideal time to measure them. And also we found out that the TEER measurement is not very gentle on the cells.
And we also saw that there's a very high variability in the assay itself, and this was really a challenge for publications.
So we found out that the xCELLigence does live continuous measurements, and for that it's just easier to figure out how the barrier, so our cells, are behaving. And it's just because it's doing like every few minutes of measurements, it's just better in comparison to the TEER, where I have to measure them maybe once or like every 10 minutes, and it's less work intensive.
And it also has a very high throughput, so we can use 96 well plates, and in a lot of different systems we cannot use this high throughput.
It was actually very easy to integrate. There is not a very complicated protocol how to do it, and it's you seed them out and then put them into the system, and that's it. Whereas in comparison to the TEER, you would need to take out the plates and then put liquid on top and then measure them and then make sure that everything stays sterile. So we're doing a cytotoxicity test. So basically what we're doing is to test out whether or not the barrier function in those specific patients who have respiratory diseases, how well it functions with different compounds. So the xCELLigence has, of course, as I've already said, it does a continuous live monitoring of the cells. It is also very sensitive, so any type of morphology change it would also pick up. So in that regard, it's a very sensitive and good tool to use for us.
And also the reproducibility is very good in between the experiments and in between the triplicate. It has this function to normalize the results, so it's very straightforward and very helpful. We have already used xCELLigence for several publications in the field of allergy, where we measured the barrier function between allergic and non-allergic patients, and we could show that they are enhancing the barrier function in culture. And to summarize, the xCELLigence is a really good tool to measure barrier function and enhances the research of mucosal epithelial cells.